2007. I was there for 20 days.
What time of year?
Yeah, in 2007 I was there. So it's very good.
Yes, Mark.
Meeting you. Thank you.
I'm happy to be here. I think I'll go without the mic if that's okay with you guys. Thank you, Anna, for inviting me. I made this presentation yesterday, so we will see how it goes. Yes. Okay, so I am here in Nepal for about 5 weeks to study a disease, mainly in domestic goats, called Peste Petit Ruminant, or PPR. There we go. I'm here on behalf of the Cornell University program called Expanding Horizons. It's quite special. As a student at Cornell University, I'm able to create a broad project. It's really totally open-ended. It just has to be in some sort of developing nation, as long as I have a veterinary contact, who I have Dr. Ikendra Shita at NPI. Through that, I can draft my program, and they will actually give me funding to come over here and study it. What I'm most interested in is conservation medicine, which is really a growing field in America. It's the idea that any way we can apply our field of science to try and save ecosystems. You might think of, wildlife veterinarian is the first thing to come to mind. Working on trauma with elephants or taking snares off of jaguars. I think those are the powerful images. What I'm really interested in is livestock, because I think there's a lot of work that can be done with agriculture, and it's just such a big player in every single country in the world. So I'm really interested in the agricultural component of conservation. Oops, not that side. So PPR, it is in the family Paramyxoviridae. It is a morbillivirus. Some of you may remember the virus Rinderpest. There's only been 2 diseases that have ever been eradicated in the world, and that's smallpox, a human disease, and Rinderpest, which is a disease of cattle. It was just such huge economic consequences for Rinderpest that it actually drove the UN to eradicate it, which is very, very rare. Clearly, only 2 diseases have ever been eradicated. Very similar. Antigenically, they're very, very similar, right? So the proteins they express on their surface match very closely. Originally, it was thought that PPR was just another lineage of Rinderpest, but they've actually shown it's a whole other genus. One other species, excuse me. Oh, not that, thank you. Sorry about this. PPR originally showed up in Cote d'Ivoire, Africa in 1942. Since then, it has spread throughout Africa, the Middle East, and Asia. Nepal right there. Different lineages have come out because of this. This is important just for PCR purposes, as well as different lineages may have certain clinical signs that manifest differently. Lineage 4 is the one that we see in India, Nepal, Tajikistan, Bhutan, so that's the one I'm actually testing for. Economic impact, that's what I'm really, really interested in. This disease has huge limitations on trade, ability for farmers to develop a herd, ability for local animal health, and ability just to get, I think, recognized as a country in the global trade. Another huge one, this bottom one, is loss of animal consumption, which is more abstract, but I think this is where maybe conservation comes into play. If people don't have the food they need to raise their family, then they're probably not going to manage their natural resources responsibly. This could mean if they don't have a large enough herd of goats, maybe they go to the forest for fuel, the forest for a source of capital. This could obviously affect downstream wildlife in result of habitat loss and encroachment. PPR is a really, really insidious virus. In naive herds, meaning areas that aren't endemic, that haven't experienced the virus before, it can come in and wipe out an entire goat herd in 10 days after the onset of fever, after incubation. In endemic areas like Nepal, it's a little lower, as low as 20% mortality rates in endemic regions. You're more likely to see goats that show clinical signs, and then maybe 6 months later they're totally fine and they have now developed an immunity. Exotic ungulates, by that comes, exotic ungulates down here. That comes back to the wildlife. A very powerful example of this recently is the Saiga antelope. You find them in Mongolia. In 2012, 200,000 of them were wiped out. This was due after necropsy and looking at post-mortem lesions. They discovered this was PPR. 200,000. There were literally piles of these antelopes dead. Mongolia has a livestock program that is really, really disorganized. Not of their own fault. It's just kind of the nature of it. There's so many livestock for farmers, so really high propensity for this to happen. Clinical signs, we kind of talked about this a little bit, but something certainly to look out for if you're ever on farms. You see discharge from the nose, the eyes, the mouth. Hyperemic gums, this means very red gums, not like the anemic gums, which are white, which you would see maybe with homuncus trichostrongyles. These are parasitic worms that can live in the intestines and actually take up red blood cells. Necrotic oral lesions, little ulcers or depressions around the mouth. So my project, unlike a lot of these other projects that are presented, which had results, mine is really just beginning. I've only been here for a week, but so far we've collected 67 samples and interviewed 22 households. I'm hoping to get about 200 samples and interview around 50 households. But the whole goal is to determine the seroprevalence in the villages that I sample around Chitwan National Park. I'm doing this through a C-ELISA reaction, so testing for actual blood samples that have the antibodies against this disease. So this means that I'm testing not for goats that are sick, but for goats that have perhaps been exposed to the disease and now have survived and have this immunity. That is me and Salendra. We are sampling a goat in Parampur, very close. And then I had the great pleasure of working with Salendra again, and then many of his intermediary students in the village of Lothar. They were very helpful. They were so engaged. It was really, really great to work with local Nepali people. And great for me to have been here and work out of the US. It's a totally different experience. Something else I would really like to look at on top of just the pure antibody results is more abstract relationships to PPR. So I'm trying to figure this out through a questionnaire that I use to interview the farmers. So this includes husbandry practice. Do they have a metal roof? Do they have a straw roof? How do they graze? Do we graze the village communally? This is a virus, so it doesn't travel, it doesn't survive long in the environment like a bacteria or a prion might. It's passed through, secretions orally or through burst secretions. Many of the animals have diarrhea. That's another way that it can be passed. So if they're communally grazing in the village, then PPR in one household can spread to the whole household, and then maybe one person takes 5 goats to graze in the jungle, and then that passes to an antelope, and then it's spreading throughout. This is why it's spread from Africa throughout the Middle East and Asia now. So these are some of the things I would like to identify. Realistically, limitations I will not be able to definitively say why, or the spread of PPR in Chitwan National Park, but it's certainly a start. Current movements are really powered by the UN. Organizations such as the FAO and OIE are huge animal health organizations internationally. Vaccine is the best method, but it is unfortunately very expensive and heat sensitive. So before implementing vaccination protocols, we need to have a better idea of surveillance, which is what I'm trying to do. It's estimated that about 1.2 to 1.7 billion US dollars is spent on PPR eradication every year. So it's a really big disease. It's going to be difficult to eradicate, but I love working here in Nepal, and I hope that I can possibly contribute something. These are some additional resources. Thank you again, Anam, for having me. If there are any questions, please feel free to ask. Thank you.
So I have one query. Done the serological test, no? Serological test for
Yes,
the confirmation of
yes.
that. Now, what you have gone for that for the identification of the antigen or the antibody in the blood sample?
The antibody.
Antibody.
So so on the ELISA test, the plates are coated with the same antibody that I would potentially find in a goat. So that means that it's competitively inhibiting anything else. So when there's no antibody, the goats have not been exposed to PPR and have not developed an immunity, then I get like a blue color. But when there is an antibody from the goat, then that outcompetes the conjugate on the coated ELISA plate, and I get pale, I get no color. So yes, testing for the antibody.
What I think so, like if you do a result test, there will be an antibody and antigen complex.
Yes,
If
yes.
that in the epitope zone, if they are linked with the antibodies, then only you can identify
Mhm.
that PPR.
Exactly,
So
exactly.
that's what he identified,
Yeah.
antibody antigen complex.
Yes.
And is the disease confined only to the goats or some other cattle also? It is-
So yeah, it's you do see it in sheep, and then as well as wildlife. They have found that pigs and cattle can seroconvert, meaning that their blood can show positive antibodies, but they don't show clinical signs, and there isn't the massive die-off. The American term for this disease is goat plague, so it literally can just wipe out goats. It's most.
The hospital is specific mainly about the goat and sheep.
Yeah.
So for a specific hospital, it is
Yeah.
PPR.
I haven't seen much sheep in Nepal, so I decided to go with goats.
Good for. Thank you very much.
Yes, thank you guys.